Good day, dear students. Today we are continuing our cycle of the lecture of molecular biology, and today we will talk about structure of plasma membrane or cell membrane, and also we will talk about membrane transport. So let's start. I remind you we said on our previous lecture that the membrane apparatus of the cell includes three main parts. It is a plasma membrane or cell membrane in other words. All parts of the cell which are located overside of the plasma membrane, and this part of the membrane apparatus is called outer complex, and part of the cell which are located between the plasma membrane and cytosol, and this part of the membrane apparatus is called inner complex. The fluid mosaic model of the plasma membrane was put forward in the early 1970s by Singer of the University of California and Nicholson of the Salk Institute. The model has evolved somewhat over time, but it still best accounts for the structure and function of the plasma membrane as we now understand them. The fluid mosaic model describes the structure of the plasma membrane as a mosaic of components, including phospholipids, cholesterol, proteins, and carbohydrates, that give the membrane a fluid character. How I said that such as a living organism, it contains three main elements: proteins, lipids, and carbohydrates. The main part of lipids in plasma membrane are phospholipids. They are similar to fats but have only two fatty acids rather than three. The third hydroxyl group of glycerol is joined to a phosphate group which is negative in electrical charge. Additional small molecules, usually charged or polar, can be linked to a phosphate group to form a variety of phospholipids. For example, this type of molecules such as choline or ethanolamine, and in this case the phospholipids will be called phosphatidylcholine or phosphatidylethanolamine. Phospholipids are described as being amphithetic, having both a hydrophobic and a hydrophilic region. Their tails, which consist of hydrocarbons, are hydrophobic and are excluded from water. Their heads, however, which consist of the phosphate group and its attachments, are hydrophilic and have affinity to water. Because of their structure, when phospholipids are added to water, they self-assemble into aggregates so that the phosphate head make contact with the water and the hydrophobic hydrocarbon tails are restricted to water-free areas, and in this case are from liposome or micelle. What are differences between liposomes and micelles? Liposomes are composed of lipid bilayer separating an aqueous internal compartment from the bulk aqueous phase. Micelles are closed lipid monolayer, only one layer, with a fatty acid core and polar surface or polar core with fatty acids on the surface, and in this case it will be called inverted micelle. It is often used in medicine in drugs manufacturing. The cavity of liposomes are filled by some medicine, for example antibiotics or hormones, and when liposomes take in the human organism, the membrane of liposomes integrated in the membrane of the human cell, and drugs enter to the cell. Another type of the lipids in the plasma membrane are steroids. Steroids include cholesterol, and certain hormones. They are characterized by a carbon skeleton consisting of four fused rings. Cholesterol is a common component of animal cell membranes and functions to help stabilize the membrane. It is also the precursor from which other steroids are synthesized. The high level of cholesterol in the blood can cause atherosclerosis. Atherosclerosis is the disease when different types of the lipid, includes cholesterol, adhere on the inner wall of blood vessels. As a result, blood flow worsens, and if it happened in blood vessels of the heart, it could cause heart attack. In brain vessel, stroke, and upper and lower extremity, gangrene. Proteins. Membrane proteins are classified into two main categories: integral proteins and peripheral proteins. Integral proteins are generally transmembrane proteins with hydrophobic regions that completely span the hydrophobic interior of the membrane. The hydrophobic ends of the molecule are exposed to the aqueous solutions on either side of the membrane. Integral transmembrane proteins may cross the membrane only once, or it may weave in and out, crossing several times. Integral monotopic proteins are permanently attached to the membrane from one side. Peripheral proteins are not embedded in the lipid layer at all. They are loosely bound to the surface of the membrane, often to the exposed part of integral proteins. The cell membrane contains not only lipids and proteins, but also carbohydrates. The carbohydrate components take the form of short polysaccharide chains, which project from the outer side of membrane. Some of the polysaccharides are attached with the phospholipids, but the vast majority are attached to the proteins to form glycoproteins. The main function of carbohydrates is identification of the type of cell. They play a key role in cell-cell recognition, ability of a cell to distinguish one cell from another, and important in organs and tissue development, basis for rejection of foreign cells by immune system. And we said that the main part of the membrane apparatus is the outer complex. I repeat that the outer complex is the part of membrane apparatus which is located outside from plasma membrane. In bacteria, it is called cell wall or membrane. In plant cell wall or cellulose. In animal cell glycocalyx. The main function of outer complex is reception and protective function. The inner complex of the cell is the region which is located between plasma membrane and cytoplasm. This area is poor in organelles and more dense than the main part of cytoplasm. There are many types of fibrillar proteins, for example spectrin and actin. They are located intercellular part of the receptors and some enzymes. The main function is structural, transport function and movement function. And now, um, I want to show you some funny cartoons. It is funny, but at the same time very scientific. So let's watch.
Interesting one. So, when you talk about a lipid in general, many lipids are nonpolar. Think of oil, for example. It's nonpolar. It won't dissolve in water. Water is polar. But a phospholipid is interesting because one part of it is polar, the head, and the other part of it is nonpolar, the tail. It's amphiphilic. Let's explain what we mean. We often refer to the polar head of the phospholipid as hydrophilic, which means that part loves water. Well, if a lipid could love. The nonpolar tails are hydrophobic. They do not like water. These phospholipids arrange themselves into a phospholipid bilayer with the nonpolar areas here in between, away from any water. It also allows this area in between to be separated from the outside and inside. Water can be found on the inside and outside areas. Also, these phospholipids, they don't just stay put. They move around. It's the fluid mosaic model after all. This gives the cell membrane flexibility. Phospholipids can even flip-flop around, but that's far less common. Remember that this entire phospholipid bilayer borders the whole cell. It would be a sphere even though we're just looking at one area of it. We have an entire video that talks about which molecules can go in and out of this membrane and which ones can't that you can view, but for now, we're going to look at some of the other structures more in depth. Cholesterol. Cholesterol often gets a bad reputation. And while cholesterol that builds up in the arteries can be a problem, cholesterol in your cell membrane is critical. If temperatures drop, the cholesterol can actually function kind of like spacers between these phospholipids, keeping them from becoming too packed. Or vice versa, the cholesterol can actually function to connect phospholipids, to keep them from becoming too fluid in warm temperatures. Proteins. In protein synthesis, we talk about why it's so important for cells to make proteins. Many proteins are found in or on the cell membrane, and they play major roles. Peripheral proteins, like the name suggests, tend to be on the peripheral area of the membrane. So while they're on exterior areas of the membrane, they generally are not going to go through the membrane. That's for integral proteins. Integral proteins go through the membrane. Oh, and peripheral proteins can sit on them sometimes. Because of location, these proteins tend to have different functions. Integral proteins, with their potential to go through the membrane, are frequently involved in all kinds of transporting methods for all kinds of materials. Some relevance? Consider the breakfast you ate this morning. Your body digested what you ate for breakfast to obtain glucose. Once in the bloodstream, those glucose molecules can't just squeeze through the phospholipid bilayer to enter all of your cells. The glucose molecules are too big and polar. But your cells need glucose to survive, to make ATP, and they rely on integral proteins to get it. Peripheral proteins tend to be more loosely attached, since they're generally not stuck in the membrane. They have an assortment of functions such as acting as enzymes to speed up reactions or attaching to the cytoskeleton structures to help with cell shape. Both protein types can have carbohydrates bound to them, which can make them considered a glycoprotein. If the carbohydrates attach to a phospholipid, you could have what's called a glycolipid. Glycoproteins and glycolipids can identify the cell as belonging to the organism, self and non-self recognition, which is very important when you're fighting pathogens. They can also be involved in many kinds of cell signaling. In fact, here's some relevance. A glycoprotein known as CD4 is found on the surface of some of your immune cells. The CD4 glycoprotein is essential for some of these immune system cells to interact with each other and activate. However, it's also exploited by the HIV virus. The HIV virus uses that CD4 glycoprotein as a way to bind to helper T cells, which it then can infect. Understanding the components of the cell membrane and how those components are involved in recognition and cell signaling is critical to understanding how to fight back against many viral and bacterial diseases. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
I hope you liked movies, and now we will continue. And second part of our lecture will be about movement in and out of the cell or transmembrane transport. As you know, there are two main types of the transport. It is a passive transport, which include osmosis and diffusion, and active transport, which can be primary and secondary. Another classification is exocytosis and endocytosis. Endocytosis includes such processes such as phagocytosis, pinocytosis, and receptor-mediated endocytosis. Now, let's talk about this transport into detail and then remind you what is the passive transport. What are the main characteristics of passive transport? It is transport without energy and along a concentration gradient. And how is that? It includes two main processes. It is osmosis and diffusion. Let's talk about osmosis. Osmosis is a spontaneous net movement of water and solvent molecules through a selectively permeable membrane into a region of higher solute concentration. With that the water tries to equalize the concentration between two sides of permeable membrane. Osmotic pressure is the head of pressure which can be developed by a solution when permitted to take up water by osmosis. When the osmotic pressure of the external solution is lower than that of the cell, the solution is said to be hypotonic to the cell. On the other hand, if the cell is surrounded by solution whose solute concentration exceeds that of the cells, water flows out of the cells, which therefore shrinks. In this case, the external solution is said to be hypertonic to the cell. If the solute concentration of the cell and its surrounding medium are the same, there is net flow in either direction, and the external solution is said to be isotonic with the cell. The osmotic flow of water into a cell is called endosmosis, and the flow out of it is called exosmosis. All this can be readily demonstrated with human red blood cells. A solution of 0.9% sodium chloride is isotonic with human cells, and if red blood cells are placed in such a solution, they will neither shrink nor swell. However, if they placed in hypotonic salt solution, they shrink perceptibly. This is known as laking or crenation. Look at the picture, crenation, or in another word, laking. If placed in hypotonic solution, they swell and may even burst, a phenomenon known as hemolysis. And let's watch another funny movie about the osmosis.
Function properly. We're going to explore what happens when you place a cell in a solution that is much more salty, hypertonic, much more watery, hypotonic, or has an equal salt concentration, isotonic. Remember that cell membranes are selectively permeable. They allow certain particles to pass through, but not others. Usually, larger molecules can't fit through the membrane without special channels. The cell membrane is fitted with special protein channels called aquaporins, which allow water molecules to pass through without expending energy. However, the ease with which the water molecules can cross is going to depend on something called the concentration. Concentration is a measure of how much solute there is per volume of solvent. In other words, whether the liquid is more salty or more watery. We'd usually expect that the molecules would follow a process called diffusion. They would flow from areas of higher to lower concentration. Eventually, they will reach equilibrium, where there will be an equal concentration on both sides of the membrane. However, in the case of a liquid like salt water, the salt molecules are much, much too big to fit through the membrane, so the only particle that can move is water. Let's look a little more closely. Water molecules are highly attracted to salt molecules. They cluster around salt and really, really don't want to move. The positive parts of the water molecule stick to the negative parts of the sodium.
Let's continue, and we will talk about osmosis in a plant cell. Plant cells generally have a solute concentration which is markedly higher than that of their surroundings. The solutes are located mainly in the sap vacuole, which is surrounded by two selectively permeable membranes, the plasma membrane and tonoplast. When the cell is surrounded by a weak solution, water is drawn into the vacuole by osmosis. The result is that the cell swells. However, it does not burst because of the cellulose walls which, once fully stretched, resist any further expansion of the cell. As water flows into the vacuole by osmosis, an internal hydrostatic pressure develops which presses the protoplast against the cell wall. This is called turgor pressure, and it is opposed by the back pressure exerted by the cell wall against the cell contents. Shrinkage of the protoplasm from the cell wall is called plasmolysis. Plant cells have become target. And the second process of passive transport is the diffusion. Diffusion is the movement of particles from a region where they are at a comparatively high concentration to a region where they are at a lower concentration. Diffusion will always proceed whenever such a concentration gradient exists, and it will continue until eventually the particles are uniformly distributed through the system. In simple diffusion, the molecules move freely and unaided through the cell membranes. In facilitated diffusion, the molecules are carried across the cell membrane by globular proteins, which pick them up one side of the membranes and deposit them on the other side. The pixelated diffusion is really faster than simple. And now let's talk about next type of the transport. It is the active transport. Diffusion is a purely physical process in which molecules or ions move from a region of higher to lower concentration. Now we know it. However, there are certain biological situations where the reverse happens. Molecules or ions are moved from a region of lower concentration to a region of higher concentration. They move against a concentration gradient. This movement against a concentration gradient is called active transport. Active transport cannot be accounted for as purely physical forces. It will only take place in a living system that is actively producing energy by respiration. It has been found that variables such as temperature and oxygen concentration, which influence the rate of respiration, also influence the rate of active transport. Further evidence that active transport is linked with energy production is supplied by biochemical studies. Energy in cells comes from the hydrolysis of adenosine triphosphate (ATP). Anything that inhibits the formation of ATP or prevents it from being used stops active transport from taking place. Active transport takes place by means of carriers in the cell membrane. The carrier molecule is a protein which spans the entire membrane. The substances to be transported becomes attached to the carrier on one side of the membrane, and the configuration of the carrier molecule then changes in such a way that the substances is moved through it to the other side of the membrane. The necessary energy comes from the hydrolysis of ATP. Active transport mechanism can be divided into categories. Primary active transport directly uses a source of chemical energy, ATP, to move molecules across a membrane against their gradient. Secondary active transport, or in other words, co-transport, uses an electrochemical gradient generated by active transport as an energy source to move molecules against their gradient, and thus does not directly require a chemical source of energy such as ATP. Let's talk about primary and secondary active transport in detail. Primary active transport. One of the most important pumps in animal cells is the sodium-potassium pump, which moves ions of sodium out of cells and ions of calcium into them. Because the transport process uses ATP as an energy source, it is considered an example of primary active transport. Secondary active transport. The electrochemical gradients set up by primary active transport store energy, which can be released as the ions move back down their gradients. Secondary active transport uses the energy stored in these gradients to move other substances against their over gradients. As an example, let's suppose we have a high concentration of sodium ions in the extracellular space, thanks to the hard work of the sodium-potassium pump. If a road such as channel of carrier protons is open, sodium ions will move down their concentration gradient and return to the interior of the cell. In secondary active transport, the movement of the sodium ions down their gradient is coupled to the uphill transport of other substances by a shared carrier protein, a cotransporter. For instance, a carrier protein lets sodium ions move down their gradient, but simultaneously brings a glucose molecule up its gradient and into the cell. The carrier protein uses the energy of the sodium gradient to drive the transport of glucose molecules. And another classification of the transport through the cell membrane, it is subdivided to transport to exocytosis and endocytosis. Exocytosis describes the process of vesicles fusing with the plasma membrane and releasing their contents to the outside of the cell. Endocytosis is the process of capturing a substance or particle from outside the cell by engulfing it with the cell membrane and bringing it into the cell. Phagocytosis. Phagocytosis is a process by which a cell uses its plasma membrane to engulf a large particle, more than 0.5 micrometers, giving rise to an internal compartment called phagosome. Now let's watch this picture and let's talk about some stages of phagocytosis. The first stage, small particles are taken up by phagocytosis to form phagocytosis vesicle. The second stage, a nerve lysosome approaches the vesicle. The lysosome fuses with the vesicle and discharges its content into. The lysosome enzymes digest the particles and the products of the digestion are absorbed into surrounding cytoplasm. Digestion is complete and the vesicle moves towards the cell surface. The vacuole membrane fuses with the plasma membrane and any indigestible matter is void. And now let's talk about pinocytosis. Pinocytosis means cell drinking and is essentially similar to phagocytosis. Teeny pinocytic channels are continually being formed at the cell surface by indigenation of the plasma membrane. From the inner end of each channel small vacuoles are pinched off and this moves toward the center of the cell. Smaller vesicles may then pinch off each vacuole and this migrate to different parts of the cell. Pinocytic channels provide a means by which liquid can be brought into the body of the cell and their breaking up into numerous vacuoles aids distribution and increases the surface area across which absorption can take place. The membrane lining the vacuoles derived from the plasma membrane remains intact. Any materials absorbed into the cytoplasm from a vacuole must traverse the lining membrane first before it can be regarded as fully inside the cell. Pinocytosis is therefore not a substitute for transport across the plasma membrane, but a supplementary process facilitating it. And now let's talk about endocytosis. Endocytosis is a process which can be subdivided into groups, non-specific, which can occur in the different place of the plasma membrane in the part which is called caveola, and specific type of endocytosis, or in another word, clathrin-mediated. Let's talk some more about specific clathrin-mediated endocytosis, or in other words, receptor-mediated endocytosis. Clathrin-mediated endocytosis of many receptor types begins with the cargo ligands binding to the receptor of the cell membranes. The cargo ligand and receptors will then recruit adapter proteins and clathrin prescalerons to the outside membrane of the cell around where budding will form. Budding of the plasma membrane then occurs, forming a clathrin-created feed. Other receptors can nucleate a clathrin-coated feed, allowing formation around the receptors. A mature feed will be cleaved from the plasma membrane through the use of membrane-binding and fusion proteins such as dynein, as well as other bar domain proteins, forming a clathrin-created vesicle that then uncoats and typically fuses to a cytic endosome. Once fused, the endocytosis cargo receptor and/or ligand can then be sorted to lysosomal recycling or other traffic pathway. So, to organize our new knowledge, let's watch some finally moving to organize our new information.
No problem going through the cell membrane, and they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example. Like some gases, oxygen and carbon dioxide gas, those are great examples. This is known as simple diffusion. Also, it doesn't take any energy to force those molecules in or out, so it's known as passive transport. Simple diffusion moves with the flow, meaning it moves with the concentration gradient. Molecules move from high concentration to low concentration. So when you hear someone saying that something's going with the concentration gradient, that's what they mean. They mean it's going from a high concentration of molecules to a low concentration of molecules. Now, remember how we said the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some transport proteins act as channels. Some of these proteins actually change their shape to get things across. Some of them open and closed based on some kind of stimulus. All of these are good things because it's helping with molecules that may be too big to cross the membrane on their own, or molecules that are polar and therefore need the help of a transport protein. This is known as facilitated diffusion. It's still diffusion, and it still moves with the concentration gradient of high to low. It doesn't require energy, though, so it's also a type of passive transport. It's just the proteins are facilitating or helping things pass. Charged ions often require a protein channel in order to pass through. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel at a fast rate across the membrane, it passes through protein channels called aquaporins. So these are all examples of facilitated diffusion, which is a type of passive transport and moves with the concentration gradient of high to low concentration. Now all the transport we've mentioned has been passive in nature. That means it's going from high concentration to low concentration. But what if you want to go the other way? For example, the cells lining your gut, they need to take in glucose. But what if the concentration of glucose in the cell is higher than the amount of glucose concentration in the environment? We would still need to get the glucose in, so it's going to have to be forced against the regular gradient flow. Movement of molecules from low to high concentration will take energy because that's against the flow. Typically, it's going to require ATP energy. A reminder that ATP, adenosine triphosphate, it has 3 phosphates. And when the bond for the last phosphate is broken, it releases a great amount of energy. Yeah, so ATP is a pretty awesome little molecule. ATP can power active transport to force molecules to go against their concentration gradient. And one way it can do that is actually energizing the transport protein itself. One of our favorite examples of active transport is the sodium-potassium pump. So that's definitely something worth checking out. There's other times a cell needs to exert energy for transport. We're still in active transport right now, but let's say a cell needs a very large molecule. Let's say a big polysaccharide. If you check out our biomolecule video, that's a large carbohydrate. Well, you may need the cell to fuse with the molecules it's taking in order to bring it inside. And this is called endocytosis. Think endo for in. Often this fusing of substances within the cell membrane will form vesicles that can be taken inside the cell. Endocytosis is kind of a general term. There are different types of endocytosis depending on how that cell is bringing substances inside. Amoebas, for example, rely on a form of endocytosis. In this example, pseudopods stretch out around what they are going to engulf, and then it pulls it into a vacuole. And there are other forms too, such as the fancy receptor-mediated endocytosis. This is where cells can be very, very, very picky on what's coming in because the incoming substances actually have to bind to receptors to even get in. Or pinocytosis, this allows cells to take in fluids. So to the Google to find out more details of the different types of endocytosis. Exocytosis is the reverse direction of endocytosis. Think exo and exit. They sound very similar too. Exocytosis can be used to get rid of cell waste, but it's also really important for getting valuable materials out that the cell has made. Want a cool example? Well, think back to those polysaccharides. Did you know that large carbohydrates are also really important for making plant cell walls? Cell walls are different from cell membranes. I mean, all cells have membranes, but not all cells have a wall. But plant cells do. And if you're going to make a cell wall, you're going to need to get those carbohydrates that are produced in the plant cell out of the cell to make the wall. So there's a great example of when you'd need exocytosis right there. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, today our lecture is over. I will happy to see you in our next lecture. It will be about nuclear apparatus of the cell, and have a good weekend.